In 2019, 13.7 million people around the world died from infectious syndromes. At least 1.2 million reported people had nothing that could be done for them. We once had the capacity to prevent these needless deaths, but our ability to fight bacteria is dwindling because of the insurgence of antimicrobial resistant pathogens. Antimicrobial resistant infections are currently one of the leading causes of mortality worldwide and are projected by some to reach 10 million deaths annually if we continue as we are. This begs the question, what is being done to increase our ability to fight antimicrobial resistance? Hi folks, my name is Cole and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at the safety trial of a new antibiotic, Gepotidacin. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. There's also more information for you in the description below. Now, before the discovery of antibiotics, infections were rampant and deadly. Childbirth was extremely dangerous, and small cuts or burns could induce infections that quickly went out of control. Many plants used for millennia are known to have antimicrobial action, but the first known modern antimicrobial agent was only discovered in the 1900s. It was an anti-syphilitic drug called salvarsan. Penicillin was discovered in 1928 and was the first general use antibiotic brought to market in 1941. Now, because of how effective penicillin was at preventing infection-related death, it was termed a miracle drug. Sadly, no medicine can work miracles. This did, however, pave the way for the discovery of many new classes of antibiotics. In the following decades, antibiotics were discovered that could inhibit the synthesis of bacterial nucleic acids, proteins, and cell walls. This led to the previously unheard of ability to prevent sexually transmitted infections, decrease childhood infections, make surgery and joint replacements more safe, and even increase the lifespan and yield of livestock by keeping them healthier. However, never far away from the release of a new antibiotic was the first reports of a wild bacterial strain found to be resistant to it. Antibiotic resistance occurs through natural mutations of bacteria or the transfer of antibiotic-resistant genes. When a bacteria is resistant, it renders antibiotics useless because their activity is being counteracted. Resistance is due to multitudes of factors, which include overuse, not finishing a treatment, and poor infection control. This is proving to be a growing problem. Because antibiotics have widespread use across many different areas, antimicrobial resistance is a complex problem. The One Health approach brings together multiple sectors and stakeholders to communicate and work together in the design and implementation of programs, policies, legislation, and research to attain better public health outcomes. This kind of approach is necessary because since the 1980s, no new class of antibiotic has been discovered, and the profit margin, thus motivation for companies to discover a new antibiotic, is low. Until now. Gepotidacin was first reported in 2016, and early studies found that it was extremely effective against antibiotic-susceptible and resistant bacteria. Gepotidacin is a novel bacterial topoisomerase inhibitor, which is a fancy way to say that it inhibits the process of bacterial DNA replication. It binds to a distinct region of the bacterial machinery, which is different from where fluoroquinolone antibiotics bind to. Now, because of how effective it is against bacteria, there are multiple ongoing clinical trials with Gepotidacin. There is one phase 3 trial currently in progress for Gepotidacin ability to combat uncomplicated urinary tract infection indications. Another phase 3 trial against uncomplicated urogenital gonorrhea is also underway. When looking at the safety of these antibiotics, we need to assess how the drug interacts with us and how we interact with the drug. Pharmacokinetics is the study of how the drug interacts with us. It is centered around the principles of drug liberation, absorption, distribution, metabolism, and excretion. To fully understand this, there are some important terms to know. When a drug is first taken, it is absorbed into the body up to a maximum concentration, Cmax, which takes a certain amount of time, Tmax. Elimination of the drug from our system is identified after Cmax, by the time required for the drug to lose half of its maximum concentration, its half-life. This correlates with clearance of the compound through the kidneys, which is expelled in urine. Now, a measure called area under the curve gives us an understanding of the overall drug exposure based on the concentration over time. Depending on how a drug is delivered, like through our mouth, nose, or skin, it may affect how it behaves in our bodies. Heck, even different ways of preparing tablets are known to have different effects. Oral medication can come in a variety of formulations, like free base, high shear wet granules, or mesylate salt, each with a slight modification to the active ingredient. As clinical evaluations of Gepotidacin in adults progressed, mesylate salt oral tablets were presented for potential commercialization. With this, different formulations need to be further assessed to understand how they affect the body. This includes all of the adverse events, from gastrointestinal pain and vomiting all the way up to life-threatening instances. Now, I want to take a moment and really highlight why clinical trials of new antibiotics are so important. It is necessary to look at different formulations of a compound because they may have different pharmacological effects from what was previously studied. Furthermore, doses of increasing strength can have variable adverse effects on people because of the way the drug interacts with our bodies. With that in mind, people of all ages get infected by bacteria. Therefore, there is a need to know what doses are safe and effective for different age groups to best utilize the tools we have discovered. If you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper we're focusing on today. This paper is called Pharmacokinetics of Oral Formulations of Gepotidacin, a Triazaacenaphthylene bacterial type 2 topoisomerase inhibitor in healthy adults and adolescent participants, by Barth et al. From GlaxoSmithKline in Collegeville, Pennsylvania, USA. This paper is divided into two sections. In the first section, the authors looked at the way that the body clears different oral formulations of Gepotidacin in adults. In the second section, they looked at the pharmacokinetics of different doses of oral Gepotidacin in adults and adolescents. The first Gepotidacin studies gave people a 1500 milligram dose of either mesylate salt capsules or tablets, both of which were similar to one another under fed or fasted conditions. In the current open label study, 26 healthy adult participants were enrolled, who received 1500 milligrams of Gepotidacin in a single oral dose. This was given in the form of free base roller compacted tablets, high shear wet granulation tablets, and reference mesylate salt capsules. This was done under fasted conditions in a randomized order with a washout of 3 days. When comparing how free-base tablets compared to the reference mesylate salt capsules, the authors found that all were rapidly absorbed in the plasma, and the only major difference between the formulations was that the Cmax of the roller compacted tablet was within bioequivalence levels to the capsules, while the high shear wet granulation tablets had a higher Cmax. When looking at the adverse events experienced by all the participants, the authors found that those who took the roller compacted tablets experienced the least incidences. Of all those affected, around 50% of the participants had gastrointestinal related adverse events. Notably, 4 people experienced moderate adverse events, which was most commonly diarrhea. Thus, this data supports that all Gepotidacin formulations tested here were safe comparable to one another. In a previous Phase 2 Gepotidacin study for women with uncomplicated urinary tract infections, a free-base tablet formulation showed itself to be adequate for use as a commercial antibiotic. This led to further development and clinical evaluation of a 750 milligram oral mesylate salt Gepotidacin tablet. In the second study here, the authors performed a 2-part double-blinded study meant to investigate the pharmacokinetics of these newly formulated tablets in adults and adolescents, the latter's dosing being informed by the former's result. This was done to determine whether adolescents should be included in future Phase 3 studies. First, 14 healthy adults were assessed and were given these commercial tablets at different doses with a washout of 3 days between treatments. They received a single dose of 1500 milligrams, 2 3000 milligram doses 6 hours apart, or 2 3000 milligram doses 12 hours apart. Compared to the 1500 milligram dose, the 3000 milligram dose had maximum plasma concentrations that were at least 2.5-fold higher. The time to reach the maximum concentration was less, and because of the larger Cmax values, the area under the curve was around 4.5 times greater. While the average half-life and renal clearance rate was similar across doses, the percentage of dose excreted in the urine values was higher in the larger dose. This can be summed up by saying, the more Gepotidacin you give someone, there accumulates more of it in their bodies. The authors next looked at a group of 15 adolescents who had an average body weight of 15 kilograms less than the adults. Based on the previous data, they were given a 1500 milligram dose or the 3000 milligram dose 6 hours apart with a 7-day washout. Compared to the 1500 milligram dose, the 3000 milligram dose had a maximum plasma concentration that was approximately 2.4 times higher. It reached the concentration just a little quicker, and it had an area under the curve value that was around 5 times higher. Notably, the half-life was almost half of the smaller dose, while the renal clearance rate and urine excreted values were similar. This can be summed up by saying the more gepotidacin you give adolescents, the more accumulates in their bodies, but it breaks down quicker. Now, the real juicy part of this paper comes from comparing adolescents to adults, not just in fashion sense and temperament, but based on the gepotidacin they were given. In both doses that the groups shared, the Cmax was around 28% higher in adolescents, but did not noticeably take any longer to do so. With the 3000 milligram dose, however, adolescents experienced area under the curve values that were around a third more, a decreased drug half-life, and correspondingly increased excreted urine values. And those correlations make sense. When looking at the adverse events experienced by all participants, 69% of adults and 88% of adolescents reported them. As the dose and time between doses increased, so too did the prevalence of adverse events. Most of these occurrences were of mild gastrointestinal nature, while for 7% of adults and 42% of adolescents, these events were not so mild. The majority of these cases were vomiting, which did not affect the absorption of the antibiotic because it happened several hours after consumption of gepotidacin. This data thus indicated that adolescents could be included in future phase 3 trials because gepotidacin was safe and had a lack of severe adverse events. To quickly summarize everything altogether, the relative bioavailability data here evaluated 2 different tablet formulations that were manufactured with gepotidacin as a free base rather than a mesylate salt. Although this study was not designed to assess bioequivalence, both tablets were within bioequivalence confidence bounds, with the high shear wet granulation tablet having only a slightly higher Cmax. From a safety perspective, there was no notable safety concerns observed, and except for a few, the adverse events were mild. The second study presented here was the first pharmacokinetic and safety clinical evaluation of the final planned to be commercial gepotidacin oral tablet, not only in adults but also in an adolescent population. Adolescents experienced a greater maximum concentration compared to adults, and when given the 3000 milligram dose, adolescents had markedly higher area under the curve, a shorter half-life, and increased gepotidacin in their urine. Overall, similar acceptable adverse event profiles were observed between the dose regimens and age populations. And though there were more in teenagers, the nature and intensity were similar. Altogether, data from both studies presented support an acceptable risk-safety profile for multiple gepotidacin formulations, and the data supported including adolescents in phase 3 evaluations using the same adult doses and regimens. Not only do I think these clinical trials are exciting to investigate and learn about, they are also significant in a broader context. This information is significant because when compared to other literature, all formulations produced and maintained gepotidacin urine concentrations suitable for antimicrobial activity. The pharmacokinetics of oral gepotidacin formulations also supported the optimum timing and dose selections in the ongoing phase 3 studies. Furthermore, the safety of systemic exposure is suitable to allow for gepotidacin to be given to a younger population, supporting the inclusion of adolescents in phase 3 trials. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this information. There were increased adverse events seen in adolescents throughout this study. The authors cite anxiety for this increase. I think it's because the adolescents weigh on average 15 kilograms less than the adults, so they are experiencing more gepotidacin in their systems. So, can there be a correlation done on the data which shows whether there's an individual link between a person's weight and the dose they took and what their adverse events were? Furthermore, in the higher doses for both populations and adolescents in general, there was a higher observed Cmax. Because a high maximum concentration can lead to toxicity problems, is there a formulation of gepotidacin that could be used to blunt the Cmax and produce more long-lasting effects? The authors also only looked at immediate bodily adverse events associated with gepotidacin. However, because the gut-brain axis is so interconnected, are there other more long-term side effects that could be happening as a result of taking this antibiotic? As always, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comments section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope you enjoyed your time doing so. If you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.